This video tutorial describes how to use GCimG Software to perform manual and automated data analyses for comprehensive two-dimensional chromatography. This tutorial describes many essential features of GCimG Software, but it is only an introduction. The software is capable of much more sophisticated analyses, which are documented in the GCimG User's Manual. Part 1 of this tutorial describes how to import a 2D chromatogram to create and view an image, how to set the image colorization, and how to navigate the image. To import a 2D chromatogram into GCimG Software, select Import Image from the File menu or click the Import Image button on the top toolbar. Next, designate the data file to be imported using the File System browser. GCimG can import data from various file formats. The file name extension of the input file must indicate the file format. For example, .cdf for ASTM ANDI format. After designating the input file, click Open. Next, the import dialog provides several import parameters and options. The timing parameters, such as the duration of the modulation cycle and the sampling rate, are essential. Most chromatographic file formats provide some or all of the timing parameters, but manual specification of some of the parameters, such as the duration of the modulation cycle, may be required for some formats. After setting the import parameters and desired options, click OK. For visualization, the detector data of the 2D chromatogram is used as pixel values to create a digital image shown in the image viewport. The pixels are colorized so that the color of each pixel indicates the value. 2D chromatograms often have a large dynamic range and a fine numeric precision, so it is important that the color scale is used effectively to signify values. If the value mapping for colorization is not appropriate, some or even all of the chromatographic peaks may not be visible. GCimG has a colorization tool that is accessible from both the top toolbar and the View menu. The Colorize interface offers many built-in color maps and allows users to build, save, and reuse custom color maps. GCimG offers two methods to map values onto a color map: parametric value mapping and automatic value mapping. For parametric value mapping, the user sets the minimum and maximum values for the colorization range and then controls the mapping function with the slider. The slider allows logarithmic value mapping, linear value mapping, and exponential value mapping functions. Automatic value mapping does not require any user-specified parameters and is the simplest mode for colorization. GCimG Software supports navigation operations to pan, scroll, and rescale the image view. Some of the navigation controls are demonstrated here. The cursor mode palette on the far left of the second toolbar allows the analyst to choose among several modes for interacting with the image. The leftmost cursor mode button with the magnifying glass icon is for view mode. In view mode, the analyst can pan or scroll across the image by click and drag with the mouse left button. In view mode, to select a region for viewing, click and drag with the mouse right button. Then, the selected region is resized to create the new image view. This resize operation changes both the scale and the aspect ratio so that the indicated region fits the viewport. The top toolbar has 4 navigation controls. The scale control consists of a box for the image scale factor and a pull-down menu which has several preset scale options. Choosing Fit View changes the scale and the aspect ratio to fit the entire chromatogram to the viewport. Typing a scale factor in the box or choosing one of the preset scale factors changes the image scale factor. The Reset button with the red square icon resets the scale to 1, resets the aspect ratio to that of the chromatogram, and repositions the image to the lower left corner of the viewport. The forward and back arrows on the toolbar allow the analyst to move forward and backward in the sequence of views, just as a web browser's forward and backward arrows navigate a sequence of web pages. The Navigator tool, which is accessible from both the toolbar and the View menu, presents a thumbnail view of the full image, with a box that outlines the region shown in the viewport. A new viewport can be set in the Navigator by click and drag with the mouse right button. In the navigator, the viewport can be repositioned by click and drag with the mouse left button. Part 2 of this video tutorial describes how to process 2D chromatograms to correct the baseline, detect and identify chemical peaks, make other analytical determinations, and save analytical results and summary reports. The first step in processing this chromatogram is to correct the baseline. Typically, chromatographs generate data values that are larger than 0, even if no compounds are being detected. In GCimage, the data value and retention times for the current cursor position are shown on the left of the status bar at the bottom of the view pane. In this chromatogram, moving the cursor over the region that is acquired during the void times of the second column chromatography shows that the data values are approximately 12 picoamps, even though no chemical peaks are present. In order to accurately quantify detected compounds, the baseline should be subtracted across the chromatogram so that the values are near 0 in void regions. In GCimage software, baseline correction is accessible from the toolbar and from the filter menu. After baseline correction, moving the cursor over the void region shows that the corrected baseline is near 0. After baseline correction, the next step is to detect chemical peaks. A blob is a set of neighboring pixels that form a single peak in the data. Although a blob is indicative of a chemical peak, coelution may cause two chemical peaks to appear as a single blob, or noise may cause a single chemical peak to appear as multiple blobs. GCimage automates blob detection to find and delineate 2D peaks in the data and provides tools for interactive corrections. This tutorial demonstrates automated blob detection. Tools for correcting errors that occur in automated blob detection are described in the GCimage User's Guide. Blob detection is accessible from the toolbar and from the filter menu. As with most operations in GCimage, important parameters of blob detection can be configured. The blob detection configuration settings are detailed in the GCimage User's Guide. After blob detection, GCimage highlights detected blobs in one of several ways. As shown in this example, each blob can be highlighted by a bubble with an area proportional to the blob volume, which is computed as the sum of the blob's data values. The configuration menu has a blob display tab for controlling blob display settings. Other blob highlight options include thin outline, thick outline, fill, and box. The blob display configuration also has controls to set the colors for blob highlights. The analyst can create and interactively switch between multiple blob display configurations. For example, between one blob display configuration with bubbles and one blob display configuration with thin outlines. In the viewport, typing in the T key will cycle between blob display configurations. GCimage also provides mouseovers that display blob attributes when the cursor hovers over a blob. The specific attributes that are shown in the mouseover are configurable. The blob mode button set on the cursor mode palette has several selectable modes for operating on blobs. This video tutorial illustrates two blob selection modes, point blob selection and polyline blob selection. To select a single blob by point blob selection, position the cursor within the blob and click the mouse left button. Then, the selected blob is highlighted as shown here with the yellow bounding box. With the blob selected, clicking the mouse right button displays the blob metadata tool. The blob metadata tool lists computed features of the selected blob such as area, which is the number of data points in the blob, volume, which is the sum of data values in the blob, peak or apex value, which is the largest data value in the blob, and the column 1 and column 2 retention times of the peak pixel. Traditional 1D chromatograms are one-dimensional functions, so a 1D peak's retention times footprint is its width and a 1D peak's summed values is its integrated area. 2D chromatograms are two-dimensional functions, so a 2D peak's retention times footprint is its area and a 2D peak's summed values is an integrated volume. Therefore, the amount of a compound detected in a 2D GC peak is indicated by the blob's volume, whereas the blob's area is just the size of its footprint in the retention times plane. The blob metadata tool also displays metadata attributes of the blob, such as compound name, chemical group name, inclusion status, and internal standard status. These attributes can be set manually by the analyst or automatically during template matching. This example chromatogram will be analyzed for benzene, toluene, ethylbenzene, and xylenes. For each of these compounds, the analyst sets the compound name and group metadata. Setting the include flag indicates that each of these compounds should be included in the report. The blobs included for reporting are indicated with a different color, which is user configurable. Manually setting the metadata for each chemical peak of interest can be laborious, but manual analysis is required only once for analytical method. Then, the same data analysis can be repeated automatically. As shown later in this video tutorial, GCimage software can capture the metadata in a template, which then can be used to automate analysis of subsequent data. One of the peaks in this data is for internal standard 2-hexanone. For this blob, the analyst sets the internal standard flag. Internal standard blobs are indicated with a different color. Associations between the included blobs and their internal standards are shown by connecting lines. If multiple internal standards are used, the pairwise associations can be changed by the analyst. Multiple blobs can be selected simultaneously. In blob point selection mode, Control-click adds or removes the indicated blob to or from the selection. Other blob selection modes are available for easily selecting multiple blobs. In the blob polyline selection mode, clicks of the mouse left button set consecutive vertices of the polyline selector, and double-click of the mouse left button terminates the polyline selector. Then, any blob intersected by the polyline is selected. This example shows the selection of the double-ring aromatic compounds. Note that the blob selection polyline is transient and disappears after it is used to select blobs. Persistent graphical objects can be drawn in graphic mode, which is described later in this video tutorial. With multiple blobs selected, clicking the mouse right button displays the Multiple Blobs Metadata tool. The Multiple Blobs Metadata tool allows simultaneously setting or unsetting metadata of multiple blobs. In this example, the Multiple Blobs Metadata tool is used to set the chemical group of the selected blobs. These compounds need not be reported individually, so their include flag is not set. The computed features and metadata attributes of each blob are summarized in the blob table. The blob table is accessible from the toolbar and the view menu. Each row of the blob table summarizes one blob. Each column of the blob table shows one computed feature or metadata attribute. The blob table can be configured to show any of dozens of computed features and metadata attributes. Clicking with the mouse left button on a column header in the blob table sorts the blobs using the indicated feature or attribute. Clicking with the mouse right button on the column header for a numeric feature allows control of the decimal precision shown. Clicking on a row in the blob table selects the blob in the viewport. Control-click selects multiple rows, and click then shift-click selects a range of rows. Clicking on Metadata Attribute field in the blob table allows editing of the metadata. The blob table can be written to a file by clicking the Save button on the blob table toolbar, or by selecting the Save Blob Table from the File menu. The blob table is saved as a spreadsheet, which can be opened in Microsoft Excel and other spreadsheet software. The Graphic Mode button on the Cursor Mode palette has several selectable modes for drawing, selecting, and editing persistent graphical objects, such as points, polylines, rectangles, and polygons. Graphics are useful both for visual highlighting and for analytical operations. After selecting the Draw Polygon mode, clicks of the mouse left button set consecutive vertices of the polygon, and double-click of the mouse left button closes the polygon. This polygon outlines the aromatic compounds with 9 or more carbon atoms. Graphics in the image are selected by point and click in the Select Graphic mode. Vertices of selected graphics are shown with small squares. Multiple graphics can be selected with Control-click. All graphics can be selected by typing Control-A in graphic mode. With one graphic selected, clicking the mouse right button displays the Graphic Metadata tool. The Graphic Metadata tool lists metadata attributes of the graphic object, including name, inclusion status, and text annotation flag. This polygon is named to indicate that it encloses the C9 Plus aromatics. The include flag is set so that the graphic is included in analytical reports. The add text object is set so that the graphic name is displayed in the image. The blob set table is accessible from the toolbar and the View menu. Each row of the blob set table summarizes one blob set. The blob set table describes two types of blob sets: group blob sets and graphic blob sets. The group blob set consists of all blobs with the same group name. A graphic blob set consists of all blobs indicated by a graphic. Selecting a row in the blob set table selects all of the blobs in the blob set. Control-click selects multiple rows, and click then Shift-click selects a range of rows. The blob set table can be written to a file by clicking the Save button on the blob set table toolbar, or by selecting Save blob set table on the File menu. The blob set table is saved as a spreadsheet, which can be opened in Microsoft Excel and other spreadsheet software. GCimage can generate a summary report for an analyzed chromatogram. The summary report can be configured with any of several sub-reports, including the chromatogram metadata, an image of the chromatogram, the blob table, and the blob set table. To create a summary report, choose Save Summary Report from the File menu. The summary report is exported as an XML file, which can be viewed on a web browser such as Microsoft Internet Explorer, or as an XLS file, which can be opened in Microsoft Excel and other spreadsheet software. Part 3 of this video tutorial describes how to build and save a template for automated chemical analysis. The blob metadata graphic objects and other determinations made by an analyst for a chromatogram can be copied to a template and then matched and applied to automatically analyze new data. To copy blob metadata to the template, set the cursor mode to blob mode and select the desired blobs. Then, click the Add to Template button. The operation is confirmed by a pop-up. To copy graphics to the template, set the cursor mode to graphic mode and select the desired graphics. Then, click the Add to Template button. The operation is confirmed by a pop-up. The template mode button set on the cursor mode palette has several selectable modes for examining and operating on the current template. In the template, each prospective chemical peak is shown by an open circle at the expected retention times position. Template graphics and other template objects also are shown. To select a template object, choose Select Template Object Mode. Then, position the cursor on the object and click the mouse left button. The selected template object is highlighted. With the template object selected, clicking the mouse right button displays the Template Object Metadata tool for the type of template object that is selected. In this example, no changes to the template are required. The objects in the current template are summarized in the Template Object table. The Template Object table is accessible from the toolbar and the View menu. Each row of the Template Object table summarizes an object in the template. Selecting a row in the Template Object table selects the object in the template. The current template can be saved to a file from which it can be used to analyze subsequent data. To save a template, select Save Template from the File menu, and then indicate the folder and file name. It is also useful to save the configuration for analyzing subsequent data. The configuration can include parameter settings for operations, the blob table configuration, and the color map. To save the current configuration, select Settings from the Configure menu. Then, on any configuration pane, click the Export Configuration button. Specify the folder and file name for the configuration file, and indicate additional configurations that are to be saved. Then click Save. Part 4 of this video tutorial describes how to perform automated analysis with a template. This portion of the video repeats the initial processing sequence shown previously. First, the analyst imports the chromatogram. As shown later in this video, this processing sequence could be automated on the import dialog, but is repeated here as another demonstration. After importing the chromatogram, the analyst clicks the Correct Baseline button. Next, the analyst clicks the Detect Blobs button. Then, the chromatogram is ready for automated analysis using the previously saved template. To read a template into a new chromatogram so that it can be used for automated analysis, first click the Template Mode button on the Cursor Mode palette. In Template Mode, click the Load Template button, which is enabled on the second toolbar. The Load Template tool allows the analyst to preview templates by browsing template files and to load template files into the current chromatogram. When a template is loaded, it can be added to the existing template or used to overwrite the existing template. Then, the Load Template tool can be dismissed. When a template is read into a chromatogram, the chromatogram itself is not changed. Only the associated template is changed. Two additional steps, match the template and apply the template, are required to use the template for automated analysis. To match the peak pattern of the current template to the blobs in the chromatogram, first click the Template Mode button on the Cursor Mode palette. In Template Mode, click the Match Template button, which is enabled on the second toolbar. The software computes a best fit of the template peaks to the pattern to the detected blobs, subject to the template matching configuration parameters. Each match between a peak in the template and a blob in the chromatogram is highlighted graphically with the location of the template peak shown as an open circle and the location of the matched blob shown as a full circle with a connecting line segment between the template peak and matched blob. The matching between template peaks and detected blobs establishes a geometric transformation that is used to reposition the other template objects, such as template graphics and annotations, relative to the detected blobs. Unless the new chromatogram is much different than the template pattern, the default template pattern matching parameters should yield a high rate of correct matches. Typically, having more peaks in the template yields more robust matching. The final step in automated analysis is to apply the template to the data as indicated by the template matching. When the template is applied, metadata from the template peaks are copied into the matched blobs, and other template objects, such as graphics and annotations, are copied from the template into the image at their transformed positions. The fundamental steps of baseline correction, blob detection, and template matching can be sequenced automatically when importing a chromatogram. As shown here, the import dialog allows the analyst to set the configuration file, activate baseline correction, activate blob detection, activate template matching, and set the template used for automated analysis, and activate reporting and name the summary report file. Then, the automated processing and analysis takes just a few seconds. As shown in this video tutorial, GCimage Software produces an analytical report from a complex 2D chromatogram in just a few minutes. Analysis involving multiple runs such as calibration, calibrated quantification, and consensus reports are performed by GC Project, which is described in the GCimage User's Manual and in the GC Project video tutorial. To learn more about the GCimage software, visit www.gcimage.com or send an email to info@gcimage.com.